in genetic engineering, what we will be doing? We will try to alter the genetic composition. Okay, we will try to alter genetic composition. So altering the genetic composition means there we will try to change the structure of the DNA. Okay, so that is one thing. So for altering the genetic composition, there we need to have some techniques, or we need to have some technology for that. So there are certain techniques to alter this genetic composition. Okay, so those this process of altering the genetic composition is known as genome editing. Okay, genome editing means the process of changing or altering the genetic composition of an organism. That is nothing but genome editing. So genome is, again, can you say what is genome? Genome is nothing but yes, complete set of the genes of an organism. So in genome editing, we will be altering that genetic makeup. Okay, so for this process, this is a process. Okay, so for proceeding with this, or for going ahead with the process of genome editing, we need to have certain important tools. Okay, so there are certain tools with which we can change this. Okay, so if we talk from the perspective of tools required for this process, so there are certain important tools. So first and foremost thing is that we need certain enzymes called restriction endonuclease enzymes. Restriction endonuclease enzyme. So this is one important tool that we require. So this restriction endonuclease enzyme is also generally known as molecular scissors. Molecular scissors. So with scissors, what you will generally do? We'll cut something. Okay. So here, what this molecular scissor, because we are using the word molecular because we are doing at the molecule level. Okay. So this molecular scissor, the function of this molecular scissor called restriction endonuclease enzyme is to cut the DNA at specific sites. So wherever it required, there it goes and it cuts the sequence of the DNA. Okay, so cutting of the DNA at the specific sites or at the required sites, that would be the function of this restriction endonuclease enzymes. Okay, so this is one thing. Then now assume that we have already cut a piece of DNA. Now this cut portion of the DNA has to be carried by some agent so that we can introduce into new genes, we can introduce into new, or we can club it with, we can unite it with other genes. So there would be a carrier for our vehicle to carry this cut piece of the DNA. Okay, so with this, what we will get? With this, where we will cut the DNA, so through this we will get a piece of DNA. So in order to transfer this piece of DNA to another genes, we will have something called vector. We'll have something called vector. So vector is simply nothing but it's the vehicle to carry DNA from one organism to another organism, or from one DNA to another DNA. Okay, so this is the function of vector. So here in genetic engineering, what we will use it as generally, vector means we will have some things called plasmids. We will have some things called plasmids. So again, what are plasmids? What are plasmids? Can you able to recollect anyone? Plasmids. Good, they are present in bacteria. Good, in one way, that is good. So what are they? They are found in bacteria, that's good. Now you see, generally when we are talking about the DNA of the other organisms, like what is the structure of the DNA? Double helical structure. So if you see in the DNA, so if you see in the bacteria, if this is the bacteria, so genes, sometimes the DNA of the genes will be in circular form. In circular form. These circular pieces of DNA present in the bacteria is nothing but the plasmids. Can you able to remember enough? Plasmids are nothing but this circular DNA that are present in the bacteria. Remember the structure. Okay, so circular DNA, shape of the DNA, you see, this is the circular shape. Whereas in other organisms it would be linear shape, double helical structure, so it is linear shape. Whereas in DNA it would be circular in shape. So generally this circular piece of DNA that is present in the bacteria refers to plasmid. Okay, so what we'll be doing, we'll be first cutting the specific DNA, piece of DNA, and then with the help of vectors, those vectors are known as generally plasmids. So these are nothing but these are the plasmids we will use, and they would be generally used for carrying the DNA pieces. With the help of plasmids, we will transfer the genes. That is one thing. So this is one another important word that you have to remember. So now can you able to recollect remember the function of restriction endonuclease? This is one important tool. Okay. Then now we have to transfer that. For transferring that, we will use vector. Vector is nothing but a vehicle to carry the DNA piece fragment. And then what is the vector that we will generally use in genetic engineering? Plasmids. So what are plasmids again? It is the circular piece of DNA that is present in the bacteria. Can you able to understand? Okay. So then, yes. Polynucleotide chain. Usually in simple fashion to say, DNA is made up of so many nucleotides. So in simple manner, we are saying that DNA is a chain of polynucleotides. That's okay. And so my second question was that do we change the order of the cells or we pair each other, we differentiate the cells? Okay. Okay. See, here we are using the word modification. So, modification of the structure of the DNA. It includes anything. We may add certain nucleotides. We may delete certain nucleotides. Or we can alter, or we can replace something. Okay. Addition can be there, deletion can be there, or alteration. It also includes like replacing one nucleotide with other nucleotide likewise. So it can be anything. In any process, we will change it. That's it. Okay. So likewise, it includes everything. That's why we are using the word entire modification. So we can use different things. So sometimes what may happen there, we can have defective genes. So defects in the genes can be structurally, there can be defects, and number wise there can be defects. So we can modify the structural defects, or we can modify the numerical defects likewise. So for that process, we will have something called gene therapy. So with the help of gene therapy, we will try to replace the defective genes by normal functioning genes. So that is also one of the application of the genetic engineering like this. Okay, so we will do anything. So it would be done during the process of development of the fetus. At the embryonic level, it would be done. Are you getting? For example, when we will do this kind of things, we'll do this when we are developing an organism. Like let's say, for example, we have to develop HIV resistant children, assume. So at embryonic level, so we have also done in 2018, there was an experiment that has been done with respect to this process also. So there, what has been done in 2018, so there were some scientists, Chinese scientists, who has replaced HIV resistant genes. Okay, so at the embryonic stem cells, in the embryonic stem cells. So this embryonic stem cells has led to development of twin kids. Their name was like Lulu and Nana. So they had the quality of HIV resistance. That was an experiment, live experiment that has been done. So we will do at the embryonic stage, so that once this embryo develops into organism like baby, so then obviously they will get certain characteristics. We'll do this at the embryo level. So that is one. Plasmid is bicircular DNA. That is one and the same. Plasmid is, we are using circular DNA as the plasmid. So that is one thing. They'll make sure that this process will happen. That's how they'll work. Okay. How are vector and that's what vector is, nothing but the vehicle used to carry the DNA piece from one genetic material to another. So this vehicle which we use generally as vector is nothing but plasmid. Okay, so plasmid is nothing but that vector. Yes. In with respect to diseases, that is different. With respect to diseases, that is different. So there, what we will do, we will call diseases like communicable diseases. When we call from the communicable diseases point of view, diseases will be just transmitted from one person to another person. So the vehicle that carries disease from one person to another person is nothing but vector. Like for example, mosquito would be a vector, so which brings, let's say, various diseases like, let's say, malaria, chikungunya. Vector is the vehicle, simple manner. Okay, here with respect to genetic engineering, we are calling it as vector. With respect to diseases, like we will have different vectors. Okay, so that is different. Okay, vector borne diseases means vehicle borne diseases. So there would be, it can be microorganisms, it can be insects, so there can be different vectors, vehicles to carry diseases. So their context varies. Okay, so that is one thing. Okay, so then next important tool that you have to remember is with respect to this genetic engineering is that we will have another thing called ligase enzyme. So we have restriction endonuclease. Restriction endonuclease is the molecular scissor, so which used to cut the different DNA sequences. So here ligase is also an enzyme, so it will generally act as molecular glue. So glue means gum which adds up something. So when we add glue, then it would be attached. So ligase enzyme is something that is known as molecular glue, so which will help in the process of attaching or uniting DNA fragments. Okay, so ligase. restriction endonuclease, ligase. These are two important end shapes. And then vector, vehicle to carry that thing. And plasmid. Plasmid is this circular DNA piece. Generally, plasmids would be used as vectors. Okay, that is one thing. And then, now, assume that we are doing this process. So we need to check the progress, like you were asking, like how will we come to know that whether what we have done has been done or not. So for checking or for verification purpose, we will use a sample DNA piece, okay, for verification at the end, whether intended developments have happened or not, okay. So this sample piece of DNA that we use to compare the results is nothing but something called probe, okay. Probe is nothing but this sample piece of DNA that the genetic engineers they will use to compare the final results. So probe is another thing that you have to remember. So these are some of the important tools that we have to remember with respect to genetic engineering. No, sample means it would be original organism. Original organism. Sample, we'll be picking it from original organism. And then after doing this process, we will compare that results with this piece, with the probe. Are you getting? So it is reference piece of DNA. Is that clear? So that is nothing but different tools that are required for genetic engineering. Can you be able to remember the tools?